Good afternoon, everyone. My name is Tibo. I am a PhD student at the Cell Biology of Viral Infection Lab, headed by Marie-Jo Amori at the IGC Instituto Gulbenkian de Ciência, Portugal. I'd like to welcome you to our first speaker of the day, Mikhail Savitski. Mikhail Savitski is a team leader and the head of proteomics core facility at EMBL. He has contributed enormously to the body of science. Particularly, he developed and used stability proteomics for understanding the phenomenon of aggregation and disaggregation, cell phenotyping, as well as detection of protein interactions with drugs, metabolites, and nucleic acids. Today, Savitski will be sharing with us his work titled Deciphering Cellular Phenotypes Using Biophysical Proteomics. Savitski, you might share your slide and begin your talk. Thank you very much.
All right. Yeah, so great to talk to all of you. It's great to see that there are many people here. And basically what I want to do in this 25 minutes is just give you an idea of what my lab does at EMBL and how this has relevance in microbiology in general and microbiome specifically. So broadly speaking, we study post-translation regulation. So we study protein-protein, protein-drug, protein-metabolite interactions. We do a lot of work on PTMs, protein quality control. And our unique angle there is that we do that also by developing and applying novel proteomic techniques, which enable us to obtain insights which are difficult or impossible to obtain by other means. And today I want to focus on one such technology, which is called thermal proteome profiling, which we've now developed already quite some time ago. And this is a technology which combines the principle of the previously developed cellular thermal shift with multiplex quantitative mass spectrometry in order to systematically determine protein thermal stability across the proteome. The idea is basically, it was initially developed in order to find protein-drug interactions, and in its initial form, it looked like this. We basically had living cells which were treated or untreated with the drug, and then you heated these living cells to a range of temperatures, and after that, lysed the cells, and the heating would induce aggregation of certain proteins that were no longer thermostable. So after lysing the cells and centrifuging down the aggregates, the supernatant would contain proteins that did not denature at a certain temperature. After that, we used so-called tandem mass tags to label the different fractions and then run them on the mass spectrometer. And this enabled precise comparison of the intensities of proteins across the different temperatures. And then we developed the whole suite of algorithms necessary to basically then determine the melting curves of these proteins and also compare them to each other, and in order to statistically assess whether there was difference in melting. And this was a very exciting thing because basically it's been known since the 50s that if a protein binds a ligand, it tends to become more thermostable. And by basically doing this kind of experimental setup, you could systematically investigate across a large part of the proteome which proteins interacted with the drug. So this was the first ever meltome of a human cell. So I'm right now sticking to the eukaryotic part of things. So these are HeLa cells. And you can see that proteins melted differentially across the proteome, and also that the so-called melting points, which is the point at which 50% of a protein is denatured, were quite reproducible. So kind of making it look promising that it could indeed be an analytical technique. We did also the very initial experiments with the pan-kinase inhibitor staurosporin, basically having two biological replicates where we treated cells with staurosporin. And what you can see here is the red dots are proteins which are reproducibly changing in their thermostability in these two biological replicates. And by looking closer at them, they all turned out to be kinases and the known targets of staurosporin, basically validating that this approach can indeed work. And this is just some example of how these melting curves could look like in such cases. So this was quite a long time ago, and then working at EMBL, we've very significantly improved this technology. It's now applicable to tissues. It's applicable to broadly study cellular processes. And I'll just take you a bit step by step what we did there in recent years. So first of all, we made it much more sensitive by basically rearranging how we do these experiments. So instead of comparing two different independent experiments, we now combine treated and untreated samples in the same experiment, which led to a very significant increase in sensitivity and enabled us to find novel off-targets of cancer drugs, and also basically suggesting repurposing strategies. The fact that the technology became much more sensitive enabled us to detect much smaller shifts in thermal stability of proteins. And this then becomes very exciting because in principle, whatever happens to a protein post-translationally, if it affects its folding, if it affects its interactions with other proteins, if it affects its localization, in principle should be reflected in a change in thermal stability. So by doing that, it seemed like a promising thing to investigate which proteins play a role in certain cellular processes, which are not detected by mere change of abundance, because many proteins do not change in their abundance when they're post-transcriptionally regulated. So we did this across the eukaryotic cycle, which actually revealed many new aspects of regulation of the eukaryotic cell cycle. And then, which will be the main topic of this talk, and this is a fantastic collaboration with NASA's TIPAS now over the years, we applied it to prokaryotes, and this is what I will talk about, of course, in the next slides. We also applied it to studying protein metabolite interactions, because again, with this increased sensitivity, you can detect changes in thermal stability when a protein binds a metabolite, even though the binding strength is much lower for metabolites than for drugs, but with this increased sensitivity, you could basically do that. And we also extended it to an in vivo setting, where basically a mouse or a rat, in this case, actually could be treated with a drug, then sacrificed, and then the different tissue parts sliced and heated up to range of temperatures, and then we could actually see with which proteins did the drug interact in different tissues. So basically taking this in an in vivo setting. We also did these experiments across the not the whole tree of life, obviously, but some sub-selection of the tree of life across different organisms, which also led to some novel insights about the connection between thermal stability and function. And one very recent exciting study, we also now can look specifically at post-translation modifications, in this case protein phosphorylations. We can look specifically at the specific phosphorylation site and see if it affects protein thermal stability. And this is also very exciting because there are many phosphorylation sites in eukaryotes, but also in prokaryotes. It's hard to deduce which of them are functionally relevant. And it turns out from this study that we can actually focus that those that change in their thermal stability are very likely to be functionally relevant, which is a really exciting aspect, because then one can pursue and thoroughly study their function. Okay, so from now on, I will focus on the eukaryotic part, on the prokaryotic part. This work has been done by really back then fantastic postdoc in the group, Andrei Mateos, who worked with me in NASA's, was a shared postdoc between us, and has now started his own group in Umeå. Oops. So, okay. And what Andrei wanted to do was basically to take this all really to the next level, and he wanted to combine thermal protein profiling with the reverse genetics approach, so gene knockouts, in order to characterize protein function in E. coli and to understand how the E. coli protein network is wired. And in order to do that, he selected 121 mutants from the single gene deletion library of E. coli, which were involved in diverse processes. He wanted to perturb the proteome in many different ways. This also included genes of unknown function. And then he basically set up a screen. So he took 2 clones for each mutant and performed thermal protein profiling on all these mutants, and he used this tandem mass specs. Back then it was TMT11 tandem mass specs to multiplex all these different mutants. And it is in such a way that in replicate one mutant would be appearing with 10 mutants, and in another replicate it would be appearing with 10 other mutants, to make this as unbiased as possible. And then the fold changes would be compared to the median of all the mutants, basically. This was basically, quite large undertaking, resulting in 220 plexed samples, which were fractionated into 1,200 samples in order to get high proteome coverage, and analyzed on basically high-end mass spectrometry instrumentation. And what we got out of the screen is that we measured protein abundance and thermostability across all these different mutants. And this is just to give you an idea which kind of data you get. So basically, for a certain protein, you can find out whether it's stabilized or destabilized. It can also change in its abundance, become more or less abundant, or it can be a combination of the two. And so when processing the data, we basically go step by step and first check whether there's any abundance difference between for a certain mutant, and then if there is, we correct for that, and then check if there's thermostability difference for a certain mutant. Following that, we calculate z-scores from the fold changes, and q values are calculated using lemma. And so in the end, what we had was 1,764 proteins, which were measured across all the different conditions for which we got abundance and thermostability scores, and 1,215 of these proteins were hits in terms of abundance or stability in at least one mutant. So 850 roughly were hits in abundance, and 890 were hits in thermostability, meaning that we succeeded quite well in perturbing the proteome in many different ways, because a substantial chunk of the proteome was changing under some of these conditions. What you see on the right is the heat maps from the basically abundance and thermostability changes. I mean, one point is that they're very diverse, they're very different from each other. So if you look at proteins, how they change in thermostability and abundance, there's no correlation. So these are really complementary measures. So typically proteins which change in abundance are not specifically changing in stability, and vice versa. So there's no, at least there's no correlation between them. And already from this heat map, there's really things lying on top which are just very, very clear in terms of what one can find. So for instance, zooming in on this red square here, this corresponds to mutants, iron sulfur cluster biosynthesis mutants. And in these iron cluster biosynthesis mutants, we can see that there's a very strong thermal destabilization of iron sulfur cluster binding proteins, because there is basically no iron sulfur clusters, and hence these proteins which bind iron sulfur clusters do not bind them, and are simply less stable and unfold easier when you heat them up, but they do not change in abundance at all. So this is actually quite an efficient way to mapping iron sulfur cluster biosynthesis mutants if you want to find out which those are. And then one of really most striking things in this study actually concerned essential proteins. So essential proteins that we saw were hits as often as non-essential proteins overall, but they were very rarely changing in abundance. And in general, the essential proteins in E. coli, and I think this is broadly applicable to other organisms as well, are higher abundant than non-essential proteins, which also makes sense because they're essential, and basically the cell, the organism wants to have a lot of them. So they were very often hits in thermostability, which made us think then whether this change in thermostability, whether this difference in thermostability were indicative of certain essential genes being more needed in certain different genetic backgrounds. So in order to really validate that and to dig into that, we used CRISPR interference to reduce the abundance of essential proteins in mutants where the thermostability changed or did not change. And by doing that, we could quite clearly see that proteins that were the essential proteins in mutants that were changing in thermostability were much more sensitive to downregulation by CRISPR interference, really indicating that this change in thermostability indicates that a change in their activity and that they become more necessary in a certain genetic background. So taking this a step further, we had data for basically all these proteins across all these different conditions, meaning that for each protein, we had 1,210 data points corresponding to the different temperatures across the 121 mutants. What you can then do with these vectors, which you have for each protein, is correlate them to each other and see which proteins change in their thermostability and abundance in very similar way across all the different mutants. And then the postulate would be that proteins which change in a very similar way are functionally related. This is illustrated here for RPOC and RPOB, the two RNA polymerase proteins in E. coli. As you can see, they correlate extremely well. Further, you can note here that these are essential proteins, and this correlation is largely due to differences in thermostability of these proteins. So this is one example, and when performing it across all proteins, you basically get this heat map showing here. And if you then look, if you then make receiver operator curves and see how well this recapitulates operants, protein complexes, and metabolic pathways, you see that this works fairly well. The proteins that do co-change are actually functionally related. And also in particular, looking at proteins which have high confidence string interaction, you see that there's a really nice receiver operator curve. And this is, of course, using what is known from the literature. Of course, there were many orphan genes there as well, which allowed us to postulate what could be their putative function based on their association and correlation with other proteins with known function. We also saw many different things regarding protein metabolism. So we saw many metabolic enzymes varying in thermostability, and we saw, if you cluster them, that they clustered according to the different parts of the metabolic pathways. And we wanted to basically investigate what is the relation between the levels of metabolites of the substrates of these enzymes and their thermostability. So in order to do that, we took 6 metabolites of the central carbon metabolism and looked at their levels using metabolomics in the 19 different mutants. And we basically saw that there was a good correlation between thermostability of the enzyme and the level of its substrate in the different mutants. But this was much less the case for protein abundance. When you correlate the abundance of a protein with the amount of substrate present in the mutant, there was much less correlation, and this was a systematic observation, meaning basically that when an enzyme changes in its thermostability, it's quite predictive of higher substrate availability for this particular enzyme, meaning that it engages more with its substrate. Generally, we also wanted to see whether mutants which have larger proteome changes also have more growth phenotypes. And for that, there was also a chemical genetic screen available across many different conditions. So we basically looked at if a mutant had many hits, did that mean many changes in thermostability and abundance? Did this mean that it also had more growth phenotypes? And there was actually very good correlation there, or I mean, you can judge yourself by looking at the plot. Which then made us think that basically we could use this data to also try to explain growth phenotypes by the changes in the proteome. And basically, because typically you would explain the growth phenotypes based on which mutant you're looking at, but basically wanted to go beyond the deleted gene and see what could be the reasons for a specific growth phenotype. And this actually made a lot of sense, and I'm showing you just one toy example here. So this is for the YBIB mutant, which becomes very sensitive to UV. And what you see here is actually the decrease in REC-R abundance in this mutant really correlates with high UV sensitivity compared to all other mutants present in the screen. And REC-R is known to be related to UV sensitivity, whereas YBAB was postulated to be related to UV sensitivity based on this previous observation. But what turns out to be the case is that if you supplement REC-R to this mutant, you basically rescue the phenotype. It becomes no longer UV sensitive, whereas if you add back YBAB, nothing happens. It still remains sensitive, meaning that it's not the deleted gene in this case, but it's actually the gene that changes and which can be detected from the screen by this approach. Okay, I think I'm pretty much on time. So this basically leads me to the summary. So we did some proteome profiling of more than 100 genetic perturbations. We saw that the essential proteins are mainly changing in thermostability, which basically provides the tool to investigating their role under different conditions. And we saw that we recapitulated a lot of known biology in E. coli and also found new things. And this is very exciting aspects which we're now further pursuing in gut microbes, because of course E. coli is a well-studied organism, even if there's a lot still not known there. Gut microbes are much less well-studied. And so applying this approach, and this is what we're doing right now, we're applying it in a modified version, holds the promise of revealing a wealth of information which is not known previously. And finally, we also saw these effects on the metabolite levels and correlation with protein thermostability, and showed that protein abundance and thermostability can explain growth phenotypes beyond the deleted gene. So this wraps up the talk. I would like to acknowledge my group. I need to always explain these pictures. This was not my idea, it was the idea of people in the group, and my birthday is on Christmas. It's a standing joke in the lab. And I would like to thank you for your attention. Thank you very much.
Thank you so much, Misha. It's a very nice picture of your lab. So the floor is open to questions. Do we have any question?
I have a question.
Please go.
Sorry. Yep. Hello. I wondered if you have done some kind of analysis in thermophiles about thermal stability, and if you think that there is a way of trying to detect structural changes in proteins that can be related to increasing the stability, if you can analyze structural data to compare thermal stability with some kind of patterns that provide this.
Yeah, both excellent questions. So in the melt matters, we did analyze thermophilus, and generally it is much more thermostable. The proteome is much more thermostable, as to be expected. So it's thermostable at much higher temperatures. Which in that paper also did an experiment where we expressed many proteins from thermophilus in E. coli, just to check that it was not the cellular environment, but really intrinsic proteome stability, protein stability, and this held up. So they were equally very much stable in E. coli. And this is basically where we stopped in that study. We didn't go beyond that. Relation between structure and thermostability, that's a super interesting point, especially now with AlphaFold, because you can predict so many things. So it's definitely something I want to look more into. We haven't done that yet so far.
Okay, thank you.
Thank you.
Do you have any more questions? So while waiting for more questions, there is one question in the chat here by Stefan Kamrad. It says, how does TPP compare with LIP?
Yeah, so this is limited proteolysis, mass spectrometry. It's also a fantastic method developed by Paolo Picotti. The methods are very complementary, and we've been talking quite some time about doing a joint study together. So basically, limited proteolysis relies on partial digestion of proteins using proteinase K, and then looking whether they're digested differently in different conditions, which can then inform about, whether a protein changed in its folding. It's a great complementary method. It has advantages in that it can potentially provide some sequence information. It works only in lysate, so as opposed to TPP, where you can work with living cells. And another really nice complementarity is that this method can provide potentially some sequence dependent information where the protein changes, whilst TPP can provide, because it looks at the whole protein, makes it actually possible to look at specific PTMs and see how they affect the thermostability of a protein. So in that, there's actually a lot of complementarity, because this is something that the other respective methods cannot do.
Okay. Thank you very much. So I think we've spent all the time now, and thank you so much,
I
Mikhail.
have one more question. Sorry, I was a bit slow in
Okay.
appearing here.
Just one last question, and then we'll move to the next speaker.
I have a question regarding the. I don't know if you hear me well because I hear some kind of.
Not so well. It's
Okay. I will write it in the chat in that case.
Okay. The question is here. So the question is how to adapt. Oh, excuse me. Is how to adapt the TPP for more complex communities, and if you think the size of database can be problematic. Note that to have the best fitting database, you sequence from the same sample.
I guess, yeah, so that's of course a relevant question when working with communities, which is, if the community gets too complex, it becomes almost impossible. I mean, if you go beyond 100 species or whatever. For a few species, like up to 5, 6, 7, 8 species, maybe even 10, I mean, you can push it quite far by fractionating the samples very deeply. It will require much more mass spectrometry time. But given that the sequences are quite different, and as long as you have the databases, but this would then be a community that you define. So you mix species together, so you know what's in there. Or you have a small community which you have sequenced, you know what's in there, so you need really to know what's in there. And then you can search a defined database, and I would say probably up to, it depends a bit at which ratios the species are mixed, but realistically, I would think maybe up to 10 different species, you could get reasonable information on you know, melting behavior of the different proteins.
Okay, thank you so much. Would like to appreciate you, Misha. Thank you so much for your talk. It was very, very educative.